Okay, let's start. Number 10. Blue Babe. The lion's tooth was still in its neck. The steppe bison had been dead for 36,000 years, frozen under Alaskan permafrost since the last ice age. And when gold miners cut it from the ground in the summer of 1979, a lion's canine tooth was still embedded in the vertebrae behind its skull. The Pleistocene steppe of Alaska bore no resemblance to the boreal tundra that covers the same territory today. It was an open, dry grassland, a cold-adapted ecosystem that extended from what is now Britain across the Bering Land Bridge to Alaska, supporting concentrations of large grazing animals that no current Arctic environment comes close to matching. Woolly mammoths moved through it. Giant short-faced bears, standing nearly 2 meters at the shoulder, occupied the same landscape. American lions, a subspecies of Panthera leo measuring roughly 25% larger than modern African lions, were the dominant apex predators on that plain. Gold miner Walter Roman discovered the carcass near Fairbanks in July 1979 while thawing permafrost with high-pressure water jets. Ardale Guthrie, a paleontologist at the University of Alaska Fairbanks, led the recovery and published the full analysis in his 1990 book, Frozen Fauna of the Mammoth Steppe. Guthrie's examination found puncture wounds on the neck consistent with canine spacing, claw rake marks across the face and shoulders, and the embedded canine tooth between two cervical vertebrae. The animal had died fighting. Its distinctive blue coloration, the origin of its name, came from vivianite, an iron phosphate mineral that had crystallized across the hide and hair during 36,000 years of chemical interaction with the surrounding soil chemistry. Beneath the vivianite coating on protected sections of skin, the original ochre-brown pigment of the fur was still visible. Internal organs remained in place. Muscle tissue had not desiccated. It had simply frozen. Guthrie and several colleagues are documented as having prepared a small portion of neck meat as stew at a dinner marking the end of the analysis project. It was described as tasting powerfully of the permafrost it came from. What the embedded lion tooth confirmed was that American lions were hunting full-grown steppe bison in Alaska well before the end of the Ice Age. Both predator and prey disappeared at the close of the Ice Age along with the grassland they depended on. The moss and shrub tundra that replaced the Pleistocene steppe stores carbon differently, releases methane at different rates, and supports a fraction of the animal biomass the grassland once did. The bison's bones showed no healed injuries from prior predator encounters, suggesting this was the attack that killed it, rather than the final assault on an already weakened animal. It died in good physical condition, in the prime of its life, in a grassland that no longer exists. The Alaskan permafrost preserved one lethal encounter in near-perfect detail. Further east, a different Pleistocene species was waiting to give back something no specimen of its kind had ever revealed before. Number 9. Sasha, the woolly rhinoceros. The horn was still attached. The woolly rhino had been dead for approximately 34,000 years, but its keratinous horn, the feature that defines its genus in the fossil record, was folded alongside the carcass when it emerged from a Siberian riverbank in 2014, intact enough to retain its original compressed structure. The woolly rhino, Colodonta antiquitatis, is well represented in the fossil record by skulls, teeth, and limb bones. It appears in the cave paintings of Chavot and Rouffignac, depicted with specificity of line that suggests the painters were working from direct observation. But the cave art is the only source of detailed physical information about the animal's exterior. Skeletal material cannot reveal hair color, skin texture, or the proportional relationship between soft tissue structures. A juvenile woolly rhinoceros with soft tissue preserved had never been recovered at all. The specimen was found in 2014 in the Saha Republic in northeastern Siberia after permafrost thaw exposed remains in a riverbank cut. Named Sasha, the animal was approximately 1.5 meters in length, indicating it was a calf less than 2 years old at death. Albert Protopopov, who leads mammoth fauna studies at the Academy of Sciences of the Republic of Saha, coordinated the recovery and initial examination.
Analysis found the hair color to be a light brownish ginger, considerably lighter than the dark brown reconstructions that had appeared in textbooks for decades, based on assumptions derived from adult skeletal proportions and environmental modeling. The horn had flattened somewhat over 34,000 years, but retained its structural layering. A second, older woolly rhinoceros, named Sparta and dated to approximately 20,000 years ago, was recovered from the same region in 2020, with orange-brown hair preserved across most of the body and confirming the lighter coloration that Sasha had first suggested. Sasha's thick, insulating fur coat was already fully developed at the age she died. Modern rhino calves are born nearly hairless in tropical climates. A woolly rhino calf born in the Pleistocene steppe faced sub-zero temperatures within weeks of birth, and her coat was functional from the start, a developmental adaptation with no living analog. The Chauvet cave paintings, dated to approximately 36,000 years ago, show woolly rhinoceroses with distinctive dorsal humps, facial proportions, and horn curvature. Sasha's soft tissue profile is a closer match to those paintings than any skeletal reconstruction produced in the 20th century. The humans who made those images in southern France may have produced more accurate depictions of the animal than the scientific community managed with 2 centuries of bone analysis. Siberia held the infant rhino for 34,000 years. Preserved in the same permafrost zone, a different discovery from 2015 would rearrange a century of assumptions about what one of the Pleistocene's most iconic predators actually looked like.
Number 8. The cave lion cubs. They looked like domestic kittens. 2 cave lion cubs dead for more than 50,000 years were so completely preserved that the texture of their paw pads was intact under examination. Their whisker follicles were visible under magnification. Their closed eyelids showed individual lashes. The cave lion, Panthera spelaea, is known from skull fragments, isolated teeth, and cave paintings. Adult specimens estimated from skeletal remains to have reached 300 kilograms or more appear in the paintings at Chauvet, Font de Gaume, and Lascaux, depicted hunting in coordinated groups which no other evidence of their behavior had confirmed. Almost nothing was known about cave lion development from birth through early life because no juvenile specimen with soft tissue had ever been recovered from any site anywhere in the world. The first 2 cubs, named Uyan and Dina by researchers, were found in 2015 in a Siberian riverbank on the Uyandina River in the Sakha Republic. A second pair was recovered in 2017 at a nearby location. Love Delen at the Center for Paleogenetics at Stockholm University led the genomic analysis, with results published in a 2021 paper confirming the animals as cave lions and placing the age of Uyan and Dina at approximately 50,000 years, and the second pair at around 28,000 years. All 4 cubs had died within the first 2 weeks of life. All 4 retained fur, retractable claw sheets, and complete facial structure. The coloration of the cubs' coats was a uniform pale buff brown, notably lighter than the tawny coat of modern African lion cubs, and without the spots that living lion cubs carry for camouflage during the first months of life. Cave lion cubs apparently were born without spotted camouflage. Their paws were disproportionately large relative to their bodies, consistent with the eventual adult size that skeletal evidence had established for the species. The overall appearance was, as Delen's team noted in published commentary, close enough to modern lion cubs that the distinction was not immediately obvious. The cave paintings depicting adult cave lions at Chauvet, made approximately 36,000 years ago, show animals without manes, a detail historically interpreted as either artistic convention or an indication that male cave lions were naturally maneless. The cub's coat structure, pale and dense rather than mane-bearing, is consistent with an animal that developed no mane at any age. The Paleolithic humans who painted those animals in southern France were painting from direct observation of a species they knew well.
The cubs preserved in Siberian permafrost confirmed the accuracy of their record. The same permafrost that held those lions had preserved something in its deeper layers that complicated any straightforward definition of what preservation means.
Number 7. The 32,000-year-old seeds. The fruit was 32,000 years old. It had been cast by a ground squirrel in the Siberian permafrost during the Pleistocene, sealed under frozen ground before the squirrel could return. And a team of researchers grew a living flowering plant from its preserved placental tissue in 2012. Ground squirrels on the Siberian steppe have been caching seeds, bulbs, and fruit in burrows for millions of years. During the Pleistocene, those burrows were sealed by permafrost before the animals can retrieve their stores, freezing the cached plant material in conditions cold enough to prevent decay, but not so destructive as to rupture every cell. Fossil burrow caches containing plant material are known from dozens of Siberian sites. The assumption for the most of the 20th century was that the biological material in those caches was irreversibly dead, structurally intact under the microscope, but no longer functional at the cellular level. David Galinsky at the Pushino Scientific Center led the team that isolated viable placental tissue from fruits of Silene stenophylla, a small Arctic flowering plant that still grows across the Siberian tundra, recovered from a frozen burrow on the Kolyma River at approximately 38 meters below the permafrost surface. Radiocarbon dating placed the material at 31,800 years old, plus or minus 300. In a 2012 paper published in the Proceedings of the National Academy of Sciences, Galinsky's team reported that cells extracted from the frozen placental tissue were successfully cultured in the laboratory, developed into complete plants, grew to maturity, flowered, and produced seeds viable enough to germinate in a second generation. A 32,000-year-old seed cache had been converted into a reproducing plant population. The resurrected plants were measurably different from their modern counterparts in one anatomical feature. The petals were narrower, with a slightly different lobing pattern at the tip. The difference was not large enough to place the Pleistocene population in a separate species classification, but it was large enough to be recorded as a quantifiable evolutionary shift across 32,000 years. The permafrost had preserved a snapshot of what Silene stenophylla looked like before 32,000 years of selective pressure, climate shifts, and population movement had altered its morphology. The culture lines established from the Pleistocene placental tissue are maintained as living specimens. They are the oldest germinated plant material in the scientific record by tens of thousands of years. The permafrost layer that held them is thawing across a region that contains thousands of similar squirrel cache sites, most of them unlocated, unsampled, and now draining into rivers. Each meter of permafrost loss represents plant genetic material from the Pleistocene that no collection effort can recover after the fact. The permafrost preserves seeds that still carried the capacity for life. In its deeper layers, it preserves something that the researchers who found it were careful to contain before they confirmed what they had. Number 6. The ancient viruses. The virus was 48,500 years old. When a research team in Marseille added it to living cells in a laboratory dish, it infected them. It had been frozen in Siberian permafrost since before anatomically modern humans left Africa, and it worked on the first attempt. Permafrost covers approximately 25% of the northern hemisphere's land surface. In the Siberian interior, it extends to depths exceeding 1,500 meters and has remained continuously frozen in the deepest sections for more than 1 million years. The upper layers hold megafauna specimens from the Pleistocene. The deeper layers hold microorganisms and viral material that predate the woolly mammoths by orders of magnitude. Before 2014, whether those ancient viruses could still infect anything was an open question in the literature, rather than an established fact. Jean-Michel Clavieri and Chantal Aubergel at Aix-Marseille University
published the first direct experimental evidence in 2014 in the Proceedings of the National Academy of Sciences, when they revived Pythovirus sibericum, a giant virus extracted from 30,000-year-old Siberian permafrost, and confirmed it could infect amoeba cells in laboratory culture. In a 2022 paper published in the journal Viruses, Clavieri's expanded team reported the revival of 5 additional ancient viral families from permafrost samples taken from multiple depths and ages. The oldest specimen dated to 48,500 years. All 5 viral strains infected amoeba. Clavieri's team used only virus families known to infect single-celled organisms rather than mammals, eliminating direct public health risk from the laboratory procedure itself. The viruses in Clavieri's studies belong to a group called giant viruses, characterized by unusually large genomes and complex capsid structures. They cannot infect humans or other vertebrates, but the permafrost archive is not limited to giant viruses. Boris Revich and Marina Podolnya at the Russian Academy of Sciences published a 2011 paper arguing that thawing permafrost could expose preserved anthrax spores and other pathogenic material from historical burial sites and infected animal carcasses. In 2016, a child died in the Yamalo-Nenets region of Siberia from anthrax traced to the thawed carcass of an infected reindeer frozen since 1941. Not prehistoric, but a direct confirmation of the mechanism the literature had been describing theoretically. Clavieri has stated in published interviews that the permafrost samples his team collected came from lakes and rivers where meltwater already drains into the open environment. The 48,500-year-old virus that infected amoeba in his Marseille laboratory had spent longer frozen than the entirety of human prehistory. Its infectious machinery was intact. The only reason it had not contacted a host in that time was the temperature of the ground it was sitting in. That temperature is rising. The permafrost contains things that are still functional after tens of thousands of years in the cold. One specimen frozen in the same Siberian ground left researchers unable to answer a question that seemed like it should have been simple.
Number 5. Dogor. Its genome could not answer the question. The puppy had been dead for 18,000 years, frozen in Siberian permafrost with its face intact, its fur present, and its teeth visible in a preserved muzzle. And when researchers sequenced its complete genome, they could not determine whether it was a wolf or a dog. The domestication of the gray wolf into the domestic dog is one of the most thoroughly studied transitions in animal evolution. Genetic analyses place the divergence somewhere between 15,000 and 40,000 years ago. Competing studies have proposed East Asia, Europe, and Central Asia as the geographic origin. The underlying biological mechanism remains debated, whether humans deliberately selected tame individuals or whether a subset of wolves self-selected toward human proximity by exploiting camp refuge and gradually lost their wariness across generations. A specimen physically dated to the middle of that estimated domestication window, with a complete genome preserved in frozen tissue, offered the possibility of resolving at least the timing question. The puppy was found in 2018 by researchers from the Yakutian Academy of Sciences in collaboration with the Center for Paleogenetics at Stockholm University in permafrost near the Indigirka River in Siberia. Radiocarbon dating placed the animal at approximately 18,000 years old. Love Dahlin at the Center for Paleogenetics led the genomic analysis, with preliminary findings published in 2019. The puppy was male, had died at approximately 2 months of age, and the tissue quality was sufficient for complete whole genome sequencing, an unusually high level of preservation for a specimen of that age. The name given to the animal was Dogor, meaning friend in the Yakut language, chosen in part as an acknowledgement of the question the genome raised. The sequenced genome placed the animal at a position in the wolf-dog phylogenetic tree that sits before the clear separation of the two lineages as they are defined by modern populations. Dogor is not a modern wolf.
it is not a modern dog. It occupies the space that the common ancestor of both would occupy, either a very early domestic dog before selection had proceeded far enough to leave a genetic signature, a wolf from a population closely ancestral to the domesticated line, or a member of a now extinct canid population that never gave rise to either modern wolves or dogs. Dahlen's team stated that additional ancient genome data from specimens dated across that 15,000 to 40,000 year window would be required to position Dogor definitively. 2 months is the age at which modern domestic dog puppies begin to respond to human social cues differently from wolf puppies of the same age, making eye contact, following pointing gestures, approaching strangers rather than retreating. Whether Dogor's behavioral profile had already crossed that threshold is something no genome can currently resolve. The question of when a wolf stopped being a wolf and became something else remains open in the scientific literature, with Dogor sitting at the center of the uncertainty. Dogor's identity remains unclassified between two categories that matter deeply to the history of the human-animal relationship. The next entry frozen in the same region carried no identity question at all, but what had happened to her before she died had no clear explanation either.
Number 4. Yuka. The wounds on the teenage mammoth were not consistent with any natural predator. The cuts were too precise at the joints. The long bones had been fractured in patterns that matched intentional processing for marrow extraction, and the animal had died approximately 39,000 years ago, during a period when the archaeological evidence for human presence in northeastern Siberia was sparse at best. The woolly mammoth, Mammothus primigenius, is the most densely documented large animal of the Pleistocene. Its teeth, tusks, and bones appear in sites across Eurasia and North America. The relationship between early humans and mammoths is confirmed through ivory carvings, bone tools, and cave paintings at sites from Spain to Siberia. What had not been clearly established for northeastern Siberia specifically was whether humans were actively hunting mammoths there in the Middle Pleistocene, or simply processing carcasses of animals that had died from other causes. Yuka was found in 2010 on the coast of the Laptev Sea in the Sakha Republic, exposed by coastal erosion. She was female, estimated at 6 to 9 years of age at death, and was recovered with unusual completeness, skin, muscle tissue, and wool covering most of the carcass. Her brain was also preserved, making her the first woolly mammoth from which a brain could be successfully recovered for analysis. Semyon Grigoriev coordinated the initial examination. A 2016 paper in Quaternary Science Reviews, co-authored by Alexei Tikhonov at the Zoological Institute, described cut marks on the bones consistent with lithic tool use, not superficial scavenging marks, but systematic disarticulation at the major joints, in sequences that indicate deliberate processing for usable meat and marrow. If the butchering interpretation is correct, it places anatomically modern humans working mammoth carcasses in northeastern Siberia 39,000 years ago. The archaeological record for human presence in that region at that date is thin, a handful of sites, none of them unambiguously establishing a hunting rather than scavenging relationship with megafauna. Critics of the hunting hypothesis have argued that large felids, particularly cave lions, could have produced similar disarticulation patterns, and that freeze-thaw cycles over 39,000 years may have distorted the bone fracture evidence. The debate has not been settled. What is not in dispute is that Yuka's preserved brain, the first intact mammoth brain ever analyzed, provided cellular tissue for examination 39,000 years after a teenage mammoth died in circumstances that no one has conclusively explained. Whatever agency was responsible for her death, the permafrost preserved its work in recoverable detail. The same permafrost on an island in the Laptev Sea preserved a different mammoth, one whose biology, rather than whose death, refused to follow any precedent. Number 3. The Maly-Yahulovsky mammoth. The blood was liquid. When researchers cut into the carcass on Maly-Yahulovsky Island in 2013, blood pooled in cavities within the surrounding ice.
The mammoth had been dead for somewhere between 15,000 and 43,000 years, frozen in permafrost through multiple decades of Arctic winter, and its blood was still fluid at a recorded temperature of -17 degrees Celsius. Blood does not remain liquid below zero under normal conditions. The protein hemoglobin that binds oxygen in red blood cells begins to degrade within days in unfrozen tissue, and the physical structure of blood, including its cellular components, is disrupted by ice crystal formation during freezing. Preserved mammoth tissue had yielded cellular material from several Siberian specimens in the early 2000s. Fluid blood, retaining mobility at sub-zero temperatures in a carcass tens of thousands of years old, had no precedent in any permafrost specimen of any species. The female mammoth was recovered by a team led by Semyon Grigoriev, which described the find publicly in 2013. Workers extracting the carcass from the permafrost using picks noted dark fluid pooling in natural voids within the ice surrounding the body, cavities that had apparently held liquid continuously despite the ambient temperature. The blood appeared deoxygenated, but was demonstrably mobile. A molecular explanation had already been established by Kevin Campbell at the University of Manitoba, whose team published a study in Nature Genetics in 2010, reporting that synthesized mammoth hemoglobin, reconstructed from ancient DNA, released oxygen to cold muscle tissue at low temperatures far more efficiently than modern elephant hemoglobin. The protein structure allowed it to deliver oxygen to working muscles even when those muscles were at temperatures that would leave elephant blood too tightly oxygen-bound to function. The biochemistry was specific, measurable, and unlike anything in any living proboscidean. The liquid blood from the Molly Yaholovsky specimen has been preserved for ongoing molecular analysis. Its existence confirmed in physical form what Campbell's genetic reconstruction had already predicted, that mammoth blood was not simply elephant blood in a cold-adapted body, but a biochemically distinct fluid optimized for a temperature range that no living elephant can tolerate. The specimen's soft tissue has contributed material to multiple cloning feasibility assessments, including work connected to Colossal Biologics, founded in 2021 with the explicit aim of producing a cold-adapted elephant-mammoth hybrid capable of surviving on the Siberian steppe. The habitat that mammoth blood was optimized for still exists. The Siberian steppe is still cold. The grasses that fed Pleistocene herds still grow there. The case against de-extinction is no longer primarily ecological. Every year, the technical case narrows. The permafrost on Molly Yaholovsky Island preserved mammoth biology at a depth that no other specimen had reached. In a glacial gully in the Alps, ice had preserved something older in human terms, and more precise.
Number 2. Ötzi the Iceman. He had eaten a full meal less than an hour before he died. The Copper Age man had been lying face down in an alpine gully since approximately 3300 BCE, frozen into the ice at 3,210 meters elevation on the border of what is now Austria and Italy, and his last meal, red deer, ibex, einkorn wheat, and bracken fern, was still in the upper portion of his digestive tract when forensic analysts examined him in the 1990s. The Alps of the late Copper Age were not wilderness in any meaningful sense. Transhumance, the seasonal movement of livestock between valley pastures in winter and high alpine meadows in summer, was a well-established practice across the region. Copper tools and polished stone axes appear in contemporary burial sites throughout northern Italy and southern Austria. Trade goods moved through the high passes. Ötzi, estimated at approximately 45 years of age at death, was not an unusual visitor to this elevation. Arthritis in his knees, hips, and spine, healed fractures in multiple ribs from earlier injuries, and deep calluses across his feet described the man who had spent decades moving through mountainous terrain. Hikers Helmut and Erika Simon discovered the body on September 19, 1991, projecting from a glacial gully in the Ötztal Alps. Konrad Spindler at the University of Innsbruck published the initial assessment in 1993.
In 2001, radiological examination revealed a flint arrowhead lodged in Ötzi's left shoulder. Albert Zink at the URI Research Institute in Bolzano, leading forensic analysis, published in 2007 in the Journal of Archaeological Science, confirmed that the arrowhead had penetrated deeply enough to sever the subclavian artery, a wound that would have caused death from blood loss within minutes. The trajectory of the arrowhead indicated it was shot from behind and below. Ötzi had been murdered. He was not fleeing when he died. His last meal, analyzed in a 2018 paper in Current Biology by Frank Meixner at URI Research in Bolzano, contained approximately 46% fat by caloric content, a deliberately high-energy preparation consistent with someone provisioning for a hard climb, not someone running. Ötzi carried a copper axe with a U-handle, one of the oldest complete copper axes ever recovered. DNA analysis published in 2012 identified 19 living genetic relatives of Ötzi concentrated in the Tyrol region. His male lineage had survived in the same geographic area across 5,300 years of recorded history. His stomach contents, his clothing made from the hides of at least 5 different animal species, and copper axe still in usable condition beside his body all survived intact.
His killer is unknown.
Whoever shot Ötzi knew exactly where to find him, approached close enough to make an accurate shot, and left the body where it fell. The oldest unsolved homicide in recorded human history has been cold since 3300 BCE, in every sense. The alpine ice preserved one man's last hours with forensic precision. In the permafrost of northwestern Siberia, a different kind of preservation had captured something with no parallel in the entire fossil record, and no comparable specimen has been found since.
Number 1. Lyuba. Her eyelashes were present. The baby woolly mammoth had been dead for 41,800 years, held in a bend of a Siberian river by silt and bacteria that sealed the carcass before decomposition could begin. And when reindeer herder Yuri Hudi found her on a sandbar of the Yuba River in May 2007, her eyelashes were still attached to closed eyelids. Most permafrost mammoth specimens are partial, exposed sections of skin, fragmented fur attached to bone, desiccated tissue on skulls or feet. The preservation of internal organs, stomach contents, airways, skin across the full body, and surface features down to individual hair follicles represents a categorically different level of survival. It requires not just cold, but an anaerobic burial environment that prevents even the bacterial decomposition that normally precedes freezing. The odds against it are difficult to calculate, because specimens preserved at this level are measured in single digits across all of recorded paleontology. Hudi alerted local authorities after his family found the carcass. The specimen was transported to the Shamanovsky Museum in Salikard before being released for international study. Daniel Fisher at the Museum of Paleontology at the University of Michigan led the primary analysis, published across a series of papers from 2009 through 2014. Fisher's examination determined that Lyuba was female, approximately 30 to 35 days old at her death, and had died by suffocation. Fine silt was present in her airways and upper stomach, indicating she had been submerged in mud, likely stepping into a muddy riverbank and sinking before she could free herself, with her mother unable to pull her clear. The preservation followed from the burial environment. Lactic acid produced by bacteria in the anaerobic silt effectively pickled the carcass before microbial decomposition could advance, then created conditions hostile to further bacterial activity. The cold did the rest. Her stomach contents showed she had ingested her mother's feces, a behavior recorded in modern elephant calves, used to acquire the gut bacterial communities required to digest vegetation, since calves were born without them. At 30 to 35 days, Lyuba had not yet begun eating solid food. Her milk tusks were just beginning to form beneath the gum line. Jessica Theodore at the University of Calgary published analysis in 2014 identifying the specific microbial communities present in her gastrointestinal tract, data that constitutes the only known sample of a Pleistocene mammoth's gut microbiome.
Francis Lecombat at the Museum of Natural History in Monaco conducted CT scanning of the full carcass, finding no skeletal trauma, no evidence of prior illness, and a complete internal organ arrangement consistent with a healthy calf that died quickly and was buried immediately. Lyuba is the most complete specimen of any Pleistocene mammal ever recovered. Every biological data point she has provided, microbiome composition, developmental timeline, fat deposition pattern, hair follicle density, milk chemistry inferred from stomach contents, exists in no other mammoth at comparable resolution. She died 41,800 years ago, in the first month of her life, in a river on a peninsula that is still there. The Yamal Peninsula has not moved. The permafrost that preserved her is thawing at a rate measured and published annually. Each year of continued warming increases the probability that the next specimen of this quality emerges from the ground before anyone finds it, and decreases the probability it survives the exposure intact. 10 specimens, preserved at 10 different moments across 50,000 years of ice. Every one of them was exposed not by excavation, but by thaw. The archive is not waiting to be discovered at a pace set by scientific planning. It is opening on a schedule determined by temperature. Most of what the permafrost holds has no name yet. Some of it, at 48,500 years old, is still functional. If you want to see more, click the video on screen.